All right, great. So it's a huge honor for me to be able to introduce Arjuna Subramanian today. I feel very proud of Arjuna. I feel like Arjuna really exemplifies like what Caltech is all about for many different reasons. So Arjuna came to us from Maryland and Colorado and then Princeton. At Princeton, he very uniquely did a double major kind of in chemistry and math, where he graduated summa cum laude. And already very interesting start, so pure math and chemistry. And he did a senior thesis with Martin Wurr on mass spec, and was also an avid sportsman, swordsman, and outdoorsman, which has been a theme that's continued through his Caltech life. And then he came to Caltech, and he's in the BMB program. And what I feel very proud of for Arjuna is he's used cutting edge tools. So he's really been a pioneer in this revolution in sort of like AI and transformer-based protein sequence analysis and protein design. But he's taken it in an extremely creative direction he'll tell you about today, using it as a tool to probe this sort of like vast unexplored protein universe of sequences that we don't know anything about. While at Caltech, he's also played softball. He's been an avid member of the Caltech Y. And I don't know if he's continued his swordsmanship, but he's a sportsman and he's really a treasured member of our community and our lab. So with that, I'll turn it over to Arjuna.
All right, well, thank you, Matt, for that really, really warm introduction. I'm really happy to be here. It's an honor to give this talk, and in general, I'm just really excited to tell you about how we've spent the last few years foraying into the untracked deep space of the protein universe and the kind of wild world of AI language modeling in biology more broadly. So this tableau here, despite maybe its overarching resemblance to a star chart or to some sort of pointillist masterpiece, this is actually a rendition of the known protein universe. If we add up all the genomes, all the metagenomes that have ever been sequenced, we can say with some confidence there are a few billion or so unique proteins that are documented or predicted. We assume they exist somewhere, some organism uses them for some life-sustaining purpose. And up here, each of those few billion gets one dot. And if two dots are close together, we're going to say that we think that those two are somehow more similar at the sequence level. And in big picture here, we see in this map there are regions of comparative density, some that are sparser. We can go out to the periphery and kind of get to isolated elements. But there's a critical feature that even this picture doesn't show, and that's everything that we suspect but don't actually know for sure to be there. The conceivable protein universe is simply enormous, almost beyond the point of comprehension. And that's because we're given by biochemistry these 20 amino acid letters. And even if I want to make a small protein domain, say just of length 100, these 20 letters give us choices that explode with combinatorial scaling. That would give us around 10 to the 130th options, just random strings if we wrote them out. Our kind of few billion are still dwarfed if we cut this down to the 10 to the 75th of these options that we could physically make if we were willing to use up all the mass in the universe, and still dwarfed by when this falls down to 10 to the 40th for the 4 billion years of evolutionary time that we've been allotted. Right, so those are the few billion that I talked about that are in these databases. And I'm not saying there's nothing special about these few billion. Of course, there is. They reflect selection for function on various timescales for things like binding nucleic acids, for sticking to the right primordial cofactors, for doing chemistry that you need to sustain life. But I want to ask you today, can it really be the case that nature has exhausted all viable, plausible, useful proteins? And I'd say the answer is a pretty definitive no. And one reason for that is that decades of laboratory work, decades of computational protein design work has yielded this menagerie of maybe functional sparks in the darkness is the way to think about it. This covers new sequences, new structures, new functions, things like environmental sensors that tell you that there's been a change in temperature. It covers therapeutic binders that stick to one cell type over another. It covers some enzymes that make and break some truly bizarre-looking small molecules. And the rate of this discovery through design process, it's accelerated year over year pretty steadily. So clearly there's untapped diversity here outside of known proteins. But we just saw we have this 20 to the n scaling problem. So it's hovering over us, and how can we go about searching through space for real proteins when we have probably a lot of junk and a lot of noise swamping out the signal? Well, one way that we could go about this would be to zoom in and look in the gaps. Look for things that are functional, but they're near proteins that we already know pretty well. So maybe they're just a few mutations away from natural neighbors. Now, I don't want you to be fooled into thinking that this solves that combinatorial scaling problem. It doesn't. If n equals 5 and we go 5 steps away, we still have 3 million options. If we go up to 10 steps, now we're talking about 10 trillion, and only a fraction of those still are going to be viable. But if we're stuck having to screen these in the laboratory, work at the bench, put some grad students through a lot of pain, then it probably still serves us to concentrate our exploratory resources when they're limited near kind of the proteins in nature that we know all too well. And this is precisely how directed evolution works. And if you haven't heard of directed evolution, in directed evolution, you take usually a natural template, you mutate kind of at random, you climb up these local fitness hills, and you find proteins that are near natural ones. And directed evolution is amazing at doing this. It's wildly successful in practice, especially for things like finding novel-to-nature chemistry. This is why Caltech's own Francis Arnold deservedly won the Nobel Prize in Chemistry for this in 2018. And I bring up directed evolution because it's landmark. I bring it up for the science, but also because it sets the table for us, really, for this idea of language-informed protein discovery. And that's because, as John Maynard Smith pointed out in this very seminal 1970 paper, and we're talking here about text for a minute, English text, not proteins. We'll come back to proteins. But Maynard Smith told us that valid words tend to lie near other valid words in space, connected by paths that go through other valid words. If you've ever played with one of these word ladders over here on the left, we can change one at a time and go from shine all the way to snarl. 2 words, they're real. Every word in that ladder, I hope, is a real English word. And they're connected by paths of valid mutated words. Okay, a little bit of a mouthful there, but the point is that directed evolution exploits the exact same thing, just for proteins. So, in these notions of paths, of connectedness, this idea that we have this confidence that we're going to find sort of functional density of proteins near other functional proteins, and then we concentrate our efforts there in the immediate vicinity at the fringes of natural protein space. But more likely is not the same thing as only. And where I want to really challenge everyone today is with the idea that we don't have to go 1 step at a time. We don't have to stay near the edges of nature. We can instead simultaneously alter huge chunks of protein sequence at once. And by doing that, we can still find structure, we can still find function, and we can zoom out beyond the fringes into the far-flung, outlying deep space of the protein universe. So let's do that. Let's zoom out, maybe way, way out. How on earth are we going to approach this search problem now? This is a lot of cold, dark space that we have to explore to find proteins. Well, one way we could try doing it would be to just start sampling at random. And that's fine, but our favorite combinatorial scaling problem is going to come back to bite us right away, because I can just tell you here the rule of thumb for firing at random in protein space is for every 100 billion or so that you try, you get one thing that works. And you might recall that I already told you there are only a few billion unique proteins in all of nature. So this is just not going to be sustainable. Random means we'll miss everything. Because perhaps our old friend Maynard Smith was right in his many ruminations on the nature of a protein space, that protein space looks something like this, that it's fractal-like, that it's populated by these alternate networks. We have nature here in the middle, it's this blue blob, but that there are many other options for alternate networks that are also valid. But we would never know that if we're just stuck with this kind of random sampling issue. We'd never figure this out. And what I'm going to tell you about tonight is that we can actually do better than random. There's the spoiler for you. By getting to know this kind of natural set of proteins really well, this blue blob, that we're going to learn its secrets, we'll learn its assembly rules, we'll learn its rules of protein language, and by doing that, we'll be able to forge out and discover these other alternate networks in deep protein space. And so for the rest of our time together today, I'm going to be coming back, hopefully, to kind of three frames, three motivating questions for why we care at all about finding, mapping, and characterizing the deep space of proteins. The first of these perspectives is kind of what I think of as the biophysicist's perspective. So this is the idea that there are these questions of whether there are certain protein structures that are easier to build than others. Given the 20 amino acids that we're going to stick to, given kind of our limited set of chemical constraints there, how many things do I have to worry about if I'm trying to make this beta barrel versus this thioredoxin versus this alpha helical bundle? Maybe there are some structures, we know there are some structures that are more designable than others. What are they and why? The second perspective, maybe we call this the protein engineer's perspective. And this really has to do with what useful properties I can get out of making huge perturbations to sequence. For example, maybe I could end up with an enzyme that has a kind of very new substrate preference. Maybe I could get a binder that sticks to the signature, the cell surface of only one cell type and one cell type only, and drives some signaling response that we want. Maybe it's as simple as I have a protein I like and I want to make it more thermostable, I want to make it more acid tolerant to behave under the industrial conditions that we often encounter when we're trying to scale any of these things up. In general, how can we improve and expand function by making these large sequence changes? And then the last frame, the crazy one, is the synthetic biology meets natural philosophy meets origins of life chemistry perspective. And this is very broad, but I think it works to boil this down to the question of what are the absolute minimum constraints on sequence that exist? We have our 20 amino acids. We're still going to stay with them for now, but what kind of different sets of motifs and rules can I come up with to write down proteins that are different from nature? Put another way, are there kind of alien rule sets out here for writing protein language? Can we elucidate those, and maybe those hold in radically different environments, like in a thermal spring or in a salt cavern or even on other planets and moons? And on that note, if you happen to be out in the audience, happen to work on the Europa Clipper or something, I have a collaboration proposal for you, as long as you don't think I'm insane at the end here. All right, so all these kind of framings, they're all interconnected. If we want to learn anything about any single one or all of them, we're going to have to go out into deep protein space and find more than sparks. We're going to have to find pockets of functional density. So that's where we're headed. We're going to do that with the help of some AI-powered probes, our mariners, pioneers, and voyagers of protein space in a way. So stick around for that. But first, we're going to have to step a little bit further back and set some ground rules for talking about what we mean when we talk about proteins and about sequence and structure and function and kind of the connections between them. So I would bet that if you were to think about information flow in biology, the first thing you'd probably think of is how genetic information kind of flows from DNA in the genome being transcribed to RNA being translated to protein. This is something so paramount we literally call it the central dogma of molecular biology. But each of these polymers, the DNA, our RNA, our protein, is kind of covering more complexity than meets the eye. And for proteins, we start with this amino acid sequence. And this amino acid sequence, it's written out in our alphabet of 20 letters, but that's just the first layer, officially the primary structure. Primary structure has enough information for us to specify secondary structure, so these are your elements like alpha helices, beta sheets, various loops, and also information sequence drives secondary structure to tertiary structure, this collapse into an ordered three-dimensional thing that we're showing all the way on the right. And here, we're going to keep going from structure and say that bearing in mind there's a little bit of simplification and generalization here, that structure is going to imply function, that the rich diversity that's out there in what proteins do is driven by equally rich variation in structure that's driving it. And so, this can cover everything from up top here, these beautiful horseshoe-shaped pattern recognition receptors that sit on macrophages and recognize kind of the signs of infectious microbes. This covers in the middle these selective ion channels that are responsible for maintaining membrane potential. It covers down at the bottom things like these motor proteins that are towing cargoes, things that are as massive and complicated as whole organelles to the right place in each and every one of your cells all the time. And this is just some of my favorite examples out of thousands we could choose from. But the critical message to take away from this is that structure begets this kind of functional richness. Okay, so those are the basics of information flow in protein world: sequence to structure, structure to function. And as far as protein design is concerned, there's one more feature that we have to take away about this sequence structure function map. And that's that this isn't some neat, easy bidirectional one-to-one-to-one mapping. It is true that if I start with one sequence, that sequence will code for exactly one native structure. But not so if I take a target structure. There are going to be many sequences for any of my target structures that encode it. So if I take any point, say in this blue swath over here in sequence space, we're going to expect that folds, collapses irreversibly to this beta propeller. Now, this kind of goes hand in hand, just remember, with that animating question we had about some structures being more designable than others. There's a lot of degeneracy built in to this map. And it's an empirical fact that there are lots of sequences that go to any one structure. And because of that, this is kind of where protein design got its first paradigms as well. The guiding route for a long time was to start with some target that you care about in structure space, and then look at the sequences in nature code for it, and just try to figure out the rules on your own. Write them down, stare at them, break them up, decide that you're guessing right, order a bunch of DNA, throw it into your favorite host system, and hopefully these proteins are real and there and stable and folded, and you just win. But nothing is ever that straightforward. A little bit of the time, it can be. So sometimes that approach works really well. If the structure you want is simple enough, the sequence rules that pattern it can be as well. So one classic great example of this is that if you just look at the amino acids and you take the ones that are the most nonpolar and you take the ones that are the most polar and you pattern out a sequence where you expect one type to end up on one face of an alpha helix and the other type to end up on the other, this turns out to be enough to drive these pseudo-random sequences to collapse into these helical bundles. There's only one sequence rule here. It's very simple. It's just like prefers to associate with like. And it's enough to reliably give you this very stable 4 alpha helical bundle structure. It even gets you some functional diversity, it turns out. You can have bundle proteins that are very simple enzymes. You can have bundle proteins that do things like pick up iron, things that would have been very useful on primordial Earth, things that are still very useful today, if you're a bacterium. But this is only because this is a simple structure. If we try to go any more complicated than this, the sequence rules start to get out of our control very quickly. You can still have some success sometimes. So, if you have a deep enough pool of sequences you can draw on, you can make a multiple sequence alignment, you can sample out of it, and you can get slightly more complicated but still small topologies, like this twisted beta sheet of the WW domain. But it's really, really hard, it turns out, to get all that distant from nature when you do something like this. This was a huge accomplishment at the time, the synthetic WW, until it turned out that it was 70% identical. Nothing, really, in evolutionary terms, to a real natural protein from the sea lamprey. Again, they didn't know at the time because we didn't get this genome until 15 years after the design campaign. But it underscores this huge problem that designing for novelty is just really hard. We get stuck to natural sequence space far too easily. And it turns out that like even physics-based models, and physics-based models are just so near and dear to my heart. I mean, I think Matt told you I came up as a physical chemist and as a math guy. These were supposed to be the gold standard and the best things ever. But still, it becomes almost impossible to escape kind of the gravitational pull of natural protein space. You can spend hundreds of thousands of CPU hours using one of these models. You can recombine fragments really carefully. You can try to find the right loops. You can do all these things, search for that one sequence with the right thermodynamics, the right kinetics to drive you to your target structure. And these are target structures that are actually new to nature usually. These are like structures we don't even see. And still you do it, and then you find out that some microbe in seawater or in a soil sample somewhere just already did it. This wasn't novel at all. New to us, but new to nature, not in the slightest. And as I said, this for many years really was the state of the art for protein design. These models that were almost glued to the edge of natural protein space. And this held until around 2021, which is a year when the Earth shook, cats and dogs lived together, and everything changed for good. And I'm not being that hyperbolic about what the arrival of AlphaFold meant for protein kind of science and protein design. I had a lot of snarky things to say at the time, but in hindsight, these journalists, they kind of had it spot on, more maybe than we did. We were too skeptical. I'm retelling, I think, maybe a very well-known story at this point about AlphaFold. The developers of AlphaFold over at DeepMind took home their own Nobel just this past December. But I really think it's worth noting, and it's hard to overstate the impression that they made by walking into CASP that year. This is the benchmark competition for all protein structure prediction. And they came in, and they just blew everyone out of the water. I mean, you don't even have to know how this is scored. This is AlphaFold. This is everybody else. And among other things, they became the first group, the first model to successfully predict every target in the CASP competition to not just backbone accuracy, but to passable sidechain level accuracy. This was a watershed moment. AlphaFold's results are one thing. They're a great thing. They speak for themselves. But in many regards, it's as or more remarkable just how they did it. Because in many ways, it's not actually that different from teasing information out of MSAs, what we just saw a few slides ago for that small beta sheet. The biological intuition here is essentially unchanged. It's still about learning kind of connections between sequence positions, so that's this kind of coevolutionary information in the MSA representation, learning the connections between that and structure through these local geometric constraints. And you're doing so from many related sequences at the same time. The special thing here was that DeepMind was able to hook this up to a new computational architecture. They were able to hook this up to the transformer, a piece that is great at figuring out which parts of a sequence, which parts of an MSA, which of these are important enough to pay attention to, as it were, and which ones are not, which ones can you just ignore. And so, all AlphaFold actually was two halves stuck together. This half where you have a part that is loaded up with transformers, learning kind of signal patterns and sequences, and this second part, this model that learns how to predict from the representation learned by the transformers and take it to the actual structural coordinates. Both of these parts were hard, but this was a data limitation problem, and this was a deep learning problem. And so this part is the one that we care about because we're trying to push on sequence constraints. And this was the first thing that was able to kind of take a really deep look into sequence constraints and start looking kind of beyond nature. But for the purposes of exploration, there's one thing that would be even better than what was going on in AlphaFold. And that would be if we could throw out the MSA part entirely. And biologically, that's a little bit heretical, but MSAs have been a problem for us in design. Pre-AlphaFold, post-AlphaFold, they're chaining us way more closely to natural protein space than we would like. So what if we just got rid of them? And we can do that, it turns out, and to do that, we have to lean all the way into transformers. We have to take this abrupt mental leap to treating proteins as text for natural language processing. So that's what we're going to do. We're going to take proteins, and we're going to take language modeling, and we're going to stick them together, and now we're going to have protein language models. So this is wonderful, but how does it work exactly? What's a protein language model doing, and to learn about sequence, to learn about structure, to learn about function, to learn about the connections between all those layers that we need for design? Well, simply the way this works is that language models, these pLMs, they learn kind of crucial representations about proteins by being shown a lot of natural sequences, having parts of those sequences blacked out, so these masks are blacked out positions, and then we challenge the model to predict back what it cannot see. And if you do that for millions, tens of millions, hundreds of millions of protein sequences for a lot of rounds, for a lot of GPU hours, they get really good at solving this task. And in the process of solving this task, they get an implicit grasp for structural determinants, for residues that are physically in contact. They get a grasp for maybe where ligand binding sites are. They get a grasp for where active sites are in an enzyme. And the reason that we know this is because we can peek into the innards of these models. We can look into their guts, look at the weights, look at the tens of millions to hundreds of billions of learned parameters that are there. These parameters, they're split up across stacks of transformers. Sometimes a model has as many as a few thousand transformers working together in the biggest ones. And what we can do is that we can kind of see what happens when we apply these transformers to a sequence. What regions in that sequence are lighting up? Where are the transformers telling us to pay attention? And we can scrutinize some of these patterns and say with some confidence that, okay, some of these individual transformers, like this one down here, bottom left, are picking up short-range properties like maybe things are contacting in a beta sheet. Others, like the one at top left, are picking up long-range contacts, like these kind of far apart in sequence positions, but residues that are touching in that tertiary structure. Others, which are not shown up here, can find things like where are those active sites, maybe they can map onto a hydrophobicity scale. We kind of see elements of real chemistry, real biology being picked up here, which is reassuring, though I will add in the interest of full disclosure here that the field writ large, we have no idea what most of these transformers are doing. I'm showing you the few that we can pull out and map onto our biological intuition very neatly. But interpretability is a huge blind spot and something that I'm really hoping to see change in the near future here. I think a lot of people are really invested in that because the next step here is basically to weed out where the spurious correlations are, to make sure we're not leaving design-ready information on the table. So I think that this is going to be a huge kind of point of interest for the field as we go forward, is figuring out even further what exactly are these models learning. So to really drive this analogy to text and NLP home, life is giving us the letters, our amino acids, and then these models, they're going and trying to suss out the words, the vocabulary, the semantics, the grammar. And that means that a trained language model, once it gets really good at learning sequence determinants, we can turn it back around as a generator. We can say, okay, take a blank sequence, fill it in from scratch from left to right by adding one amino acid at a time based on what you've learned. And this is less black magic than it is marginalizing repeatedly out of a high-dimensional data distribution. That's really all it is. The special thing is that we only got that distribution because transformers gave it to us. And that's a take-home lesson maybe worth pausing for, because what MSA methods were, what a lot of the history I kind of walked through was to show you that with enough blood, sweat, tears, and math, humans were good enough to look at natural sequence data and learn some patterns. Where transformers beat us, though, is in being able to consider new patterns, synthetic data, synthetic sequences, and make an accurate decision as to whether they're sensible or not. That's what we can't do, but that's what these models can. So for our purposes, this leaves us with pLMs having a basic grasp on the core features of kind of protein sequence and structure, and it leaves us with pLMs that possess this innate explorative capacity to generate artificial sequences, decide if they're plausible, and now, just like that, we're not glued to nature like we were with our MSAs anymore. So for exploration, for going out into deep protein space, what we're going to need to do is turn these language models, turn them into agents that can move through sequence space while dialing down similarity to natural proteins, while retaining some sort of structure, some sort of function. So we're going to take these leaps and bounds, we're going to make large sequence perturbations, push out into deep space while staying clear of the nonsense patches that we saw it's littered with. So we'll stay anchored to well-defined structures, and this will kind of be our guide point for this explorative process. So we devised a strategy, essentially, for this kind of exploration into protein space, and we call this strategy fold tuning. And what fold tuning is, at a very, very high level, it's drawing inspiration from yet another kind of AI concept, which is the concept of generative adversarial networks, better known as GANs. To understand fold tuning, all you really need to know about a GAN is that a GAN has two parts again. It has a generator, it has a discriminator. What the generator is doing is it usually has some pre-training behind it, and it's spitting out examples here, fake examples, that it feeds to the discriminator. The discriminator has access to real data, and based on that, the discriminator decides, hey, this thing I'm seeing, is it real or is it fake? And you reward the generator for faking out the discriminator, and you penalize it for getting caught. And what this means is that over many, many rounds of generation, as you're kind of refining your generator, what should happen is that your artificial data gets driven towards something that mimics the look and feel of the real data, but is kind of subtly, is not exactly the same. And so when we port this over to our sequence exploration problem, now we have our generator. Our generator is a language model. For everything that we do, that specific language model is literally, it's a carbon copy of GPT-2 just for proteins. And now structure prediction becomes our discriminator. So what we do is we kind of pre-train our generator on a target fold. In our example, this happens to be GPCRs, but it can be literally anything. And then we get artificial sequences out. We check that they match our target structure. That's our equivalent of fooling the discriminator. And we reward the generator with kind of more training on the sequences that are kind of known good by the structural match process. And it's the same thing as a GAN, essentially, and with one small difference, which is that we also add in kind of an extra reward to this process for looking different from natural sequences. And a little bit corny, I'd like to think of that as almost, it's like a dash of rocket fuel we're adding to the system to kind of accelerate how quickly we can take these steps through space. So we're going to fold tune for many rounds and try to get out into deep protein space this way. So we did this. We took fold tuning, we applied it to several hundred kind of target structures of interest. And these structures encompass all the major fold classes in biology. It covers small domains, it covers large domains, it covers particularly nasty, tricky things like proteins that are actually knots. And on the functional side, it also covers a lot of different classes of protein that are of interest to synthetic biologists. So this is not exhaustive at all, but some of the examples I'm choosing to highlight here for you are transcription factor DNA binding domains in blue, composable cell surface receptor domains in gold, our GPCRs that we just saw and their associated G proteins in green, and then also some small molecule, not small molecule, but sort of small antimicrobials and toxin-related drug leads in pink. And so those are just kind of specific targets of a lot of interest. And then within each of these panels represents a different number of rounds of fold tuning. And then within each panel, we're looking at two quantities. We're looking on the x-axis at, that's telling us how good a job did we do hitting to our target structure. And on the y-axis, how good were we at diverging from natural sequences? How good were we at escaping nature's gravity and getting out into protein space? So where you want to be in one of these plots is you want to be in the top right corner, where we're hitting the structure and getting away from the sequence. And I think we start to see here that as we add rounds of fold tuning, that's happening. We're getting driven towards these higher rates of structure matching, these higher rates of sequence escape, ending up in kind of this successful part of the map. And this is even easier to see if we look at a phase portrait view of what's going on. So now on the left, we're seeing essentially kind of how structure matching and sequence escape are changing for the 10 most abundant folds in nature when we fold tune. And the takeaway there is that as we go through this, we see that we get better and better at getting away from nature, and usually without losing much of anything in our ability to hit the structure. And this is really, really good because this is telling us that, hey, our pLM probes, they're actually moving, it seems, between pockets of viable unnatural protein density. If they were going off into garbage, they would maybe get to new sequences but come all the way back down to 0 in structure. That's not happening. We're staying on target, but we're getting away from nature. And we can ask then, okay, we had this question about designability, and these models that drift into the top right-hand corner, where we're hitting structure and escaping sequence, that sounds a lot like things that are easy to design. And so we can just use that to come up with a proxy metric for protein designability. And when we do that, we see that the kinds of structures that pop out in the designable corner are things like TIM barrels, beta propellers, so these very, very common enzyme folds that do a lot of diverse reactivity in nature. We see these transmembrane barrels that usually form pores and channels. We see these beta helices that are a really common part of like protein-protein interaction motifs. And what all of these have in common is that they have high degrees of symmetry, they have repeats in their structures. And so it's just interesting to note that that is what these language models seem to be very well attuned to for what's an easy structure to make. Physics-based models actually struggled to do this. These were some of the hardest targets for those like Rosetta-based methods long ago. But for some reason, fold tuning is able to pick up this kind of designable repeat structure. All right, so highly designable targets are great. Trickier ones are good as well, because that's where we get a lot of specialized function. And fold tuning can also deliver to us novel versions of more complicated proteins. So that covers now everything from a top left, these knotted structures I mentioned briefly. It covers kind of cysteine-based and histidine-based coordination chemistry for binding metals that gets preserved in these zinc finger DNA binding domains. It covers more complicated multimeric membrane channels like aquaporins, which are water pores. And bottom right, it gets us also to novel versions of non-trivial enzymes. So this is a beta-lactamase down here. Beta-lactams, many antibiotics, including penicillin, are beta-lactams. So if you hear about antibiotic resistance, this is the kind of protein often that is responsible for it. And fold tuning can give us really good-looking beta-lactamases that come with an active site, a catalytic triad. They come with a selectivity filter, but they're grafted into what otherwise is like this novel sequence scaffold that looks nothing like nature. And that's kind of the theme of all of these examples. No matter how complicated the structure we're giving fold tuning within reason, it's able to discover sequences that have little to no similarity or homology to anything out there in nature. And that means, no similarity, to be clear, to like any of those few billion sequences I told you about. So this is a very big step away from the edge of natural protein space. And this is phenomenal because it suggests that we actually are in a position to completely remove ourselves from the known protein universe and still find structured and functional proteins in abundance. Okay, so it'd be even better if we could get a deeper look into how much and what exactly is changing when we go between natural sequences and fold-tuned ones. For the moment, all we're looking at up here is a flattened, dimension-reduced version of the hidden representations out of one of these models, which put simply, if you'll indulge me, is just how are these models thinking about sequences internally? And the models think in 1280 dimensional space, but we'll stick to 2. I think it's a little more tractable. And similarly, instead of looking at all 700 targets, we're going to focus on 11 just for simplicity. So just like in the first picture of protein space we looked at together, how close 2 points here are reflects how similar they are, really how similar the model thinks they are. And whatever similarity metric these models are learning, the general kind of picture here is that sequences are largely separating by fold label. So each differently colored blob is a different fold, and then within each fold, we see our fold-tuned sequences, which are tinted lighter, drifting away from nature. So they're starting to move away from their natural parents, which is what we hoped would happen. We can zoom in on specific examples. So we go back to our trusty GPCRs, and here each extra loop of fold tuning is getting us a little further away on the sequence side relative to nature. And we see that it's also kind of coarsely preserving our structural features as well. It's not doing that perfectly. This isn't, sub-angstrom level structural fidelity. There are models out there. Those are inverse folding models that can give you that kind of perfect match. But that's not what we're going for here. In fact, one of the major goals, especially as we look towards engineering applications, look towards synthetic biology implications, is that it's good to have kind of some structural plasticity. It's good to treat our template structure as imperfect and blurry, because hopefully that's going to give us some kind of structural and functional innovation that we can come back to later. And another view of this, we can turn this over into a network view of sequence similarity. So now every sequence is a node, and your nodes are blue if you're in nature, they're pink if you're fold-tuned, and each of these pairs gets connected by an edge if they have even a passing resemblance. And by here passing generous, we say that two proteins have even light zone level similarity. So really, I think of it more almost as if two proteins aren't connected. There's no evolutionary kind of match between them at all. And even with this strict approach to putting these networks together, we can observe that our fold-tuned proteins, our fold-tuned GPCRs, they're forming new clusters of sequences that are distinct from nature. We can switch targets and do it all over again. So now we're looking at immunoglobulins, which maybe you know from their role in adaptive immunity, their role in antibody structure. And it's kind of the same story. As we add these rounds of fold tuning, we're keeping our backbone topology, we're keeping our strand connectivity, but we're moving away from natural sequence space. We construct a sequence graph exactly the same way, and this kind of, I think, reinforces the same pattern as before. We get these novel infilled clusters of kind of fold-tuned immunoglobulins. It's clearly a little trickier to separate from nature in this case compared to the GPCRs, but the immunoglobulins are an ancient, extremely diverse fold, and this is still a solid step as far as diversification. All right, so one thing we could do now is we could walk through all of these examples, go through, tell 9 more stories, but they're all the same. That would be really boring. What I'd rather do is drill down one more level into sequence and try and elucidate what new patterns are actually being learned here, what new rules are coming out to the fore in these populations of novel proteins. So one way we can kind of get a sense for this is we can look at local context. We can break our sequences into short subsequences. And if we go to subsequences of length 4, or so-called 4 grams, we do this because these are long enough that these are considered kind of characteristic signatures of individual protein families in nature. That's not enough to generate new examples of proteins because you're throwing away global context, you're throwing away global contacts, but it's enough to kind of judge whether, usually, if I give you an unannotated protein domain, hey, is this going to be a GPCR, or is this going to be an immunoglobulin, is this going to be something different altogether? And as it turns out, our fold-tuned sequences, they don't match these 4-gram signatures of the parent at all. We're looking at some of the same examples we've been looking at already, GPCRs, immunoglobulins, beta-lactamases, ancient folds like thioredoxins and SH3s, and as many as 1 out of every 6 of the words that we're looking at here are shifting in their usage. Every red dot on these plots is a protein word that is moving in its usage dramatically when we go from natural to fold-tuned. And so fold-tuned sequences here, we're seeing they have their own signature vocabularies that are completely different from what nature has done. And the last thing that I'll kind of mention about fold tuning that kind of came out as a surprise to me was, so one more thing before we kind of talk about experiments here, is that I mentioned when we talked about the fold-tuned GPCRs, we kind of are trying to embrace the structural plasticity, trying to see what benefits we get from treating natural structures as blurry templates. And what's very striking is the extent to which fold tuning is capable of structurally elaborating and kind of adding small changes and improvements to its targets. So we're looking at another example, again, here that we've already seen. This was the TIM barrel. We saw a few slides ago that it was highly designable. But what's also really interesting when we fold tune on TIM barrels, it's nature's most abundant enzyme scaffold, but in nature, every TIM barrel has 8-fold symmetry. So it looks roughly like this gold example up here. But when we fold tune on TIM barrels, we get to things with 9-fold, 10-fold, or even 11-fold symmetry, things that are not found in nature at all. And this is not the story of the physics-based models all over again, I'll just say, because now we have databases of predicted structures that we can compare to, hundreds of millions worth of them, and these actually are novel folds that don't appear there. So there's just something kind of, for us, that was very serendipitous about this, because evolution kind of does the same thing. Evolution reaches new superfamilies by trying out new structural ornamentations, new elaborations, new minimizations. And fold tuning is doing the exact same thing, just out in new corners of protein space, working with new sequence pieces and bricks. All right, and so for this last section, I'm going to talk about some experiments we did. And that's, everything to this point, I think, conceptually, intellectually, has been well and good. But in the end, what matters the most is that we need these fold-tuned proteins to be real. We need them to be folded. We need them to be stable. We need them to do something. Because, I've spent maybe 45 minutes here relying on language models, treating the output as real, but we know language models hallucinate. We have to be sure that fold-tuned language models aren't doing the same thing and making up proteins, just like ChatGPT will make up some citations for you or invent some new case law. That's not what we want to have happen. Okay, so something we could do is we could go out, we could try to solve a few thousand protein structures. That's really hard. That's not a lot of fun. And maybe we could trim that down to the most likely candidates by predicting what will express and what will be soluble. That's also a really hard problem. Those models don't work either. So we had to think like biochemists and come up with a sensitive and scalable functional assay for one of our targets. And so our target here was this protein called bar star. Bar star is on the right here in cyan. Bar star is a bacterial protein. It has exactly one job in real life, and that job is to inhibit its cognate ribonuclease, barnase in green, and it does that by sticking to it really, really tightly, one of the tightest non-covalent binding affinities known in nature. And the reason it has to do this is that barnase is a really potent secreted RNase. If you let barnase out, it will chew up all the RNA in its path. So if you try to make barnase in bacteria without its bar star inhibitor, you chew up your cellular RNA and you die. And that's not awesome if you're a bacterium, but it gives us a nice growth assay that we can play with, because now all we have to do is take our fold-tuned bar star sequences, find the DNA that codes for them, clone them into bacteria, turn them on with barnase, see what survives, and sequence it. And anything that sequences it should be a functional bar star. And we get a handful of variants, I think 11 of them, that are good enough to rescue bacteria from death by barnase. So to kind of figure out a little more insight into what's going on here, we can obtain AlphaFold predicted structures of some of our rescuer variants, and we can see what they would look like if they were bound to barnase. And we can compare that to the wild-type crystal structure that is known. So up here, the reference structure is in blue. Our fold-tuned bar star variants are in pink, I think, and barnase is in white, because, pro tip, you should not make structure figures in dark mode. But right off the bat, kind of the important thing here is that on this variant on the top left, where we see a fold-tuned bar star variant that is predicted to adopt the canonical bar star fold, the canonical bar star binding interface to barnase, and yet it has no similarity to anything in nature. So just kind of to reiterate that, like, this is a variant that performs toxic gene rescue empirically, but it has no sequence resemblance to any protein found anywhere on this planet. And so this is real function now that we've found out in the dark of protein space, only retrievable by a language model. So the rest of these here, top right, it's a similar story. A canonical variant, it's not quite as interesting because it has twilight zone resemblance to a natural counterpart. And then the ones at the bottom here are interesting as well because they seem to be preserving almost this minimal binding interface where you have a single alpha helix shoved into the binding cavity on Barnase, but these kind of hard to interpret twists and turns elsewhere in the structure. So again, we'd really like to have some semblance of what is going on here structurally. And we can do that. So for a perspective on what exactly is being preserved and modified here, we turned to an old trick, which, so I think I was hoping we wouldn't have to see MSAs again, but we do because we're using a technique called statistical coupling analysis. And the point of statistical coupling analysis is just to find us correlated positions in a protein. So that if we were dealing with natural proteins, we would say that when we see these correlated positions, that means that they're evolving together, they're drifting together. And we can do the same thing for our synthetic sequences to get a sense for whether there are kind of structural clusters, structural motifs that are similarly kind of being preserved as minimal structural constraints. And so what seems to be happening when we do this on bar star is that we actually get this preservation of these like co-evolving, like as residues kind of as if they were co-evolving at our binding interface. So this seems to be a minimal, like a bare minimal structural and functional constraint on bar star that fold tuning is picking up on while kind of freely innovating in the other 80% of the structure that's available to it. And for credit where credit is due, all of this analysis was done by a fabulous rotation student working with us right now, Sonia. All right, so that was bar star. Bar star gives us new sequence rules. It gives us minimal structure constraints. It gives us toxic gene rescue. And emboldened by that, we went after a very different binder, one that is honestly trickier and just worse to handle in every way. And that target is insulin. And this was a project that was joint work with another student in our group, Alec Lorenzo. And the point here was to design variants and mimics of insulin that bind tightly and bind specifically to the native insulin receptor. As part of this effort, Alex screened for us thousands of fold-tuning designed insulin variants using a platform where he combined phage display with next-generation sequencing to characterize binding. And if you want to learn more about that, and you really should, you should go to his defense in a few weeks. But as far as fold tuning, the punchline is that it does give us insulin variants that bind tightly and specifically to the receptor that we wanted. They're specific, they're enriched significantly over kind of off-target controls, and even though it's not coming through perfectly, I think, in these structures, because we're looking a little too far out from the active site, these fold-tuned insulins, what they're doing is they're actually, at least according to structure prediction, is making kind of new contacts to the receptor. So they're binding in modes that are different from what is seen with kind of the natural wild-type insulins. For bar star, we saw binders that seem to be doing the exact same thing as what nature does, but the fold tuning on insulin is opening for us this door towards, maybe there are new binding modes, new receptor activation dynamics that can be explored here, because the downstream signaling, ultimately, that's what we care about. Do you get to new phenotype? More so than just being sticky to the receptor. And this, you might recall again, was kind of our second animating question. We didn't forget about it at all, because now, with new binding modes, that constitutes functional expansion by virtue of making evolutionarily inaccessible steps through fold tuning. Okay, so that was the last major experimental push that I want to discuss today. Since I'm almost done, let's take stock of what we've seen today. So I told you that we were going to start by dissecting every sequence motif, the semantics, the grammar, kind of the natural rules of language out of one network of proteins, natural protein space, that we can have access to really, really easily. And that by doing so, we would be able to assess and find kind of equivalent rule sets out in deep protein space. And as much as we hoped that we could do this with biophysical intuition alone, we couldn't. It took the invention of the transformer to take this leap and make this whole endeavor possible. Transformers brought us the ability to identify and validate new kind of sequence languages consistent with biochemistry, but so distant from what we've seen here on Earth that we wouldn't have been able to kind of diagnose it ourselves from nature. We mixed protein language models, which at their core are just stacks of transformers, with the counterfeiting game played by GANs. And in doing so, we got to amino acid sequences that structurally resemble proteins we know and trust well, but that essentially constitute an alien proteome in other ways. And we called the method to do this fold tuning. And by applying fold tuning to several hundred kind of targets of interest, we gained hopefully a little bit of clarity into some puzzles that are foundational and some that are more applied. So we saw that symmetrical structures, repeating structures, seem to have some feature there that makes them highly designable, even with new sequence rules. We saw that, there are maybe many Maynard-Smithian alternate networks of proteins that use these equivalent sequence vocabularies and grammars. And we saw, too, that fold tuning cracks open a functional window towards new binding modes and new phenotypes, both in a model system and in one with insulin that's of real translational therapeutic interest. And kind of spinning off all of this, we expect fold tuning to have many more applications at the level both of individual proteins and at the level of interacting systems of proteins. One area that's particularly exciting to me is the prospect of these new signaling networks where we could boot kind of fully tunable orthogonal signal transduction cascades up, replace the protein-protein recognition domains with fold-tuned versions, replace the kinases that do phosphorylation with fold-tuned versions, and go see kind of what new phenotype space we get in terms of gene programs, in terms of response dynamics like ultrasensitivity. So this would be potentially huge for functional innovation in cellular engineering. Another direction we could go here is towards catalyzing new to nature chemistry. So we would have to do this with fold-tuned enzymes, and fold tuning already gives us kind of pools of enzyme candidates that we think could have kind of new activities for functions that are of a lot of interest in synthetic biology, things like gene editors, things like P450 variants that could do xenometabolism and break down pollutants in the environment. There are some signs here, too, that there is a decent amount of space we can sample from to make proteins more thermostable as well. So this is a really, really active area of interest, too. And lastly, I'll plant one final idea, which is that, as we saw here, that we could push on sequence constraints a lot, and we could write down totally new protein language rules. So given that, it's worth thinking about whether we can go even further, whether we can go on one hand towards adapting our models to work with new amino acids, new monomers that kind of have different biochemical properties, and that would give us access to new protein-like polymer spaces. This is not a radical idea either. This is a space that's of a lot of interest. For example, in biopharma, where they're trying to make these protein-like products for next-generation antibiotics discovery. And we could think even about going in the other direction, too, as far as sequence complexity, where we could compress the alphabet of amino acids in play to, say, match conditions that might be more representative of ancient Earth. Could we dial back and use fewer letters and find out just how little we need to do things like basic metabolism, genome maintenance, or even construct a full minimal cell? Because I think if there's one thing that this whole fold tuning journey has made kind of me grapple with about the nature of life and the nature of protein space, it's that life probably has many, many more solution sets at its disposal than we think. Our problem is we're just blind to most of them. And with that, I'm all done. I have a lot of people to thank quickly, and obviously I want to start by thanking Matt. Thank you for all your support and advice on this project over the last several years. You've taught me to kind of embrace high-risk research and what comes with it, and really encouraged me to pursue these questions about the nature of protein space instead of getting caught up in the AI arms race that sometimes can dominate this field. So I'm so grateful for that, and I wouldn't be anywhere near the scientist I am today without that. Obviously, my thanks as well to many, many, many Thompson Lab members past and present. I've listed some of the folks here who were involved in this project at one time or another, but not everyone didn't have space to do that. I think there are 30 people or so in this photo, just that I counted, and that's not even everyone either. My committee are all fantastic. This would have been way less successful without them. This project had many funding sources. We benefited from donated and subsidized compute. And currently, this work is split into a couple of preprints that you can check out if you're interested. One of these is on kind of fold tuning in general. One of these is Alex's project on binders that contains the insulin work. So I'll leave those codes up and take any questions.
Structure as a constraint for exploring the protein space, and two, for those alien sequences that serve or like fold into similar structure, what are some of the properties they have in common, even though they are vastly different in sequence? All right,
I'll answer the first one first, and then I'll probably need you to give me the second again. But as far as why do we use natural structure kind of as an orienting point, that's just because we need anchors to grab onto to move through protein space in kind of a logical way, in a way that just lets us navigate and find structure and find function. I mean, the alternative, and I think that there are models certainly that take different approaches where there's sequence only and you have a lot of internal scoring built in, maybe you have some reinforcement learning thrown in there as well, and the claim is that you can kind of push out through sequence alone, but it's really just an issue of that in practice that's not there yet. If you use a model like that and you get 1 out of every 100 sequences working, then it's just not useful as far as going that far beyond nature, because most of what you're seeing is garbage, and we're not interested in garbage, and garbage dwarfs everything that's real. So there's so much we can learn at first just by using kind of the real structures, the real motifs that we know are actual physical things as kind of reference points. And then the second question, I think you were asking about things that those kind of alien protein sequences that they have in common with each other, or that they have in common with the natural templates?
A little bit of both. Like, what common properties do they have that allow them to fold into a common structure?
Yeah, I mean that's kind of the question is what I think we have. I'm confident that what we have is evidence that these are kind of different vocabularies, different semantics, different grammars. But if you had to actually split those up and what are the individual rules, that's something that's a really fascinating question and another kind of active area of research and collaboration for us. Right now, we actually have an ongoing collaboration to try to figure out, if you pose this, say, as, so when we do like bioinformatics, right, a lot of the time, and you do like say just your favorite BLAST search, it's running on top of a substitution matrix, and what pairs of amino acids are similar. The idea there is to do that, but for sequence motifs. So like can we change this back into like a sequence rule transformation that these models are doing? I think that would answer your question. We're just not there yet. We have guesses for what's going on, but those guesses are the kind of thing that I think you make out of just first pass biochemical intuition, like, oh, you're changing one positively charged amino acid, so you're changing lysine for arginine, and vice versa. That's not, I'll say, what happens. Like, if it were that simple, I'd tell you it's that simple. So it's not just that you're exchanging the most similar amino acid pairs for each other, but we're trying to figure out the same thing. What are the substitution rules that have been learned?
Cool.
Mango.
Very interesting talk. And I have two questions about, so following up her question about, in addition to structure, which is your kind of your criteria to for sequence generation, what other properties you may have considered and why not them? Yeah.
Okay, so I think if I'm understanding the question right, it's that we used.
Consider function.
Yeah. So I think the question is, so we used structure, but why? What other things could you use instead as sort of the properties that you're trying to adhere to? Why not? Why not? Okay, so yeah, so there are a lot of properties that are desirable in proteins and in protein engineering that you could pick from and try to match them to for a method, whether that's a specific kind of reaction that you're catalyzing, so a specific function, a specific binding target, being more thermostable, having a higher melting temperature. And the reason, honestly, aside from the fact that that just would be a completely valid kind of approach to take, like if you decided you wanted to find thermostability preserving paths through protein space instead of structurally preserving paths, I think that's a completely legitimate thing to do, and I would be really curious to see what kind of structure you discover like in sequence space by doing that. In practice, the reason is also that, structure prediction methods are good, and like we trust them and the field trusts them. And so when we need a discriminator model to validate what we're doing, it makes sense to use AlphaFold or use ESMFold because you can align two structures to each other and say quickly, yes, this is a match, or no, it isn't. But our functional data that's available to us is not as deep. Like the functional models that do things like predict thermostability, for example, or predict like enzyme function, they exist, but they're not validated in practice, and there's, I think, still a lot to do there to make sure that they're accurate predictors and that they generalize outside of their training data. I mean, that's the other huge kind of, I think, elephant in the room for, why we do this the way we do it, is that if you want to use function prediction as a discriminator, today that means you're using a language model-based function predictor as a discriminator, and we're literally talking about sequences where we've rewarded a language model for kind of running off into the wilderness. So that just feels a little bit off to me, to trust a functional predictor on sequences that are completely outside kind of the distribution of its training data. And you had a second question.
Yeah, I have a second question. Maybe it's not exactly related to your work, but I was always confused, like from AlphaFold to protein language model, like what, like they cancel the MSA's step, like why and yeah, why we can cancel it?
Why does it work?
Yeah.
Why? Okay.
It still works.
So
Because since both of them use just a transformer, and yeah, so I feel like there is not too much like network architecture innovation, but it's still
Yeah, so I think the question is, again, and feel free to correct me if I'm getting it wrong, is that why can we go from AlphaFold with an MSA transformer to like ESM with ESM or ProGPT with a sequence transformer that only looks at sequence positions and not kind of alignment across related sequences? And it's different kinds of, I think sometimes this is just what happens when different kinds of tasks point people towards different biological intuition. So for AlphaFold, the task was, we want to be able to predict structure from sequence, knowing that limited in the structural coverage of the PDB, for example. So they kind of took a priori the assumption that a single sequence is not going to be enough. Like if we just go sequence to structure, it's not going to work. And for years, people have been using MSA to approach this problem because people tried to do structure prediction by identifying kind of sparse signals of coupling in MSAs as well for decades before this. And so I think the assumption was just that you needed an MSA for it to work. And it was only when kind of people started leaning fully into language modeling of proteins and using training data with the size of maybe like 50 million sequences, with taxonomic diversity and everything included, that it turned out that you just didn't need the MSA, that individual sequences were enough as long as you kind of sampled in a reasonable way from the diversity that is out there. Might not be a satisfying answer, but I do think it's accurate. Just we thought we needed the MSA until we realized that we didn't. I go all the way over first. I think I've been waiting. Yeah.
Yeah, I guess during the inference kind of stage when you're running the transformer model to create kind of novel sequences, what exactly are you using as the input to the generator?
Mm-hmm. Yeah. So the question is that what information do you give the generator before it gives you an artificial sequence? And the way we do it, because we're using PROT-GPT-2, which is an autoregressive model, so it was trained given to see tokens from left to right and then predict the next one. When it generates, we don't give it anything. We literally give it a blank seed and say, start filling it in. And so most of the time, you can go back actually to the generation slide maybe to emphasize that, is that most of the time it's going to start with methionine. That's the good to have an initiator, and then it'll add an amino acid or a subsequence of amino acids at a time, and kind of as you do that, maybe think about it as like you're sharpening the distribution in the next position. Because once you add just an M, it can still pretty much be almost everything. But that's how it's working. Is it's going left to right with no information. We're not giving it a seed. We're not giving it like a real example and asking it to append to that. That would probably shift the properties pretty significantly. That's definitely what's happened in other work. Zach, do you still have a question?
I was just going to say ESM2 is also a lot bigger than the Evo former, and so that also might be
Yeah, that's a good point. So Zach, thank you for kind of more following up to Mango's question about the MSA transformer versus the single sequence transformer. Yeah, AlphaFold was not actually that large a model compared to what we have now. I know you think a lot about that, that we have some of these huge language models out there now for proteins, like 100 billion parameters training on a server farm for 2 years. AlphaFold was actually nowhere near that. It was much smaller. It's a great point. Matt.
Last question is, it's a really beautiful talk, and as you were talking, you've done such a beautiful job framing this. I was wondering, do you think there's a way to adapt the paradigm, maybe invert it, to look for new folds?
This is something that, so Matt's question is, we used natural folds, and we used that to find novel sequences. What would we do? Could we do this instead? Could we do something else to use sequences or anything, I think, to find novel folds? And I think it's just a question of mining the signal accurately that's there. Because in these databases of artificial sequences that we've generated, we do see kind of structural elements that are either new or they're not commonly happening in nature. So one of these, the knots kind of got us on this topic, but also there are very rare secondary structure elements, things like alpha strands, and so yes, these are there. I think it's just a question of how do we learn from those few sequences, because they are comparatively scarce, and actually be able to kind of clone those into many more examples. Because with fold tuning, that works because the sequence training, the training task is what the model would do even if we weren't fold tuning. So the reward is very natural. The reward is saying, hey, these were your fake sequences that tricked the discriminator. Learn more, make your next round look like these too, but more like them. And so I think that it's trickier to find an equivalent to that reward step for rewarding having novel structure, whether that's a small element or a full structure. But I think if you have the right reward and the right penalty, then absolutely you could flip the problem. Andy?
Really cool talk. I'm curious whether or not you think that your fold tuning method is kind of limited towards proteins that strictly follow Leventhal's principle, where because like most or a lot of more complicated proteins that evolve by themselves evolve with chaperones and cofactors and other things.
Mm-hmm. Yeah, that's a really, really nice question. Thank you. This is a real biochemist question there. Does this only work for, basically, does this only work for proteins where sequence drives structure fully, where you just kind of collapse without the aid of something else to co-fold it, a chaperone to refold it, any kind of like templating there? I don't think that's I have an empirical answer for you, which is that I don't think that's a limit, just because there are kind of large enough things in our data set and things that I know need chaperones, like tubulins, that the structures sort of pass enough sanity checks. Obviously we don't know yet if they're real experimentally, but based on that, I don't think there's inherently a limitation. I do think that, in the context of like all of protein design, that is probably like one of the grand challenges, though, is just capturing something as simple as the right kinetics of folding. That's something that has plagued design for forever, for example, that even like really stable designed proteins, like thermodynamically really stable, often you get no hint of, say, like cooperativity in the folding. Like we know that's a huge driving force. I think there are even theories that was like the driving force behind the evolution of the selection of the first proteins. So I still think that that's a principle that is really kind of hanging over protein design, is capturing the folding kinetics properly. But I don't think it inherently stops fold tuning or inverse folding or anything else from kind of getting there for a more complex structure. Anything else? Shi-Chen.
Amazing talk, and I think you really demonstrated how this tool is super important for both like very practical design of proteins and also for kind of more fundamental probing to like evolution biology. So I'm going to ask two questions in that fashion.
Okay.
First is that you showed a UMAP on basically the fold tuning versus natural protein.
Mm-hmm.
So do you see that, like, let's say if a group of protein already has very diverse sequence, do you see that fold tuning has maybe a better time designing something that's out of natural? And the second question is on the other bigger UMAP that you showed us, which is kind of the universes.
Let's look at that. It's so fun. Let's just look at that one.
Yeah, exactly this one. So are we kind of finding smaller clusters using your tool around our existing universe, or do you think we're actually going to kind of without better word, like a mirror universe as well?
These are both fabulous questions. I love these so much. This is like these are kind of the big questions about what we're doing with this method. Like, how far are we getting outside of natural protein space? Are we getting outside at all? And so for the first one, you're asking about essentially, fold tuning versus natural within a single fold. Does it work better because maybe there's more diversity for some over others? Yes, like is the short answer to that. Like that's certainly it is easier, it seems, to design new examples into a protein family that is already diverse. With the Tim barrel being very designable, is that the chicken or is that the egg? Is it really, really designable because language models latch on to it so well? Or is it because, hey, evolution already makes 10% of its enzymes Tim barrels, so surely there's a lot to kind of harvest from and recombine and stitch together. So that's very valid. I think it is easier if you already have more diversity in your natural starting point. It would It would honestly be confusing if it were the other way around. As for your second question, how far did we get? For this, I think, I'll hedge a little bit only in that, we generated limited amounts of synthetic data, so a few thousand examples usually for each of these few hundred folds. So I don't think we have enough sampling there to start piecing together like sort of the more macro scale networks. But the beautiful thing about these language models is that generation, once you've trained them, is cheap. And so I think we should turn them back on and make those kind of sequence plots and network plots, and that would be the definitive answer. I think that was some of the point of trying to show you the two examples for like GPCRs versus aminoglobulins. There are some, like with the GPCRs, where I actually do think that we're moving decently far outside what's already there, and others where I suspect we're almost interpolating between subclasses, because some immunoglobulins are actually like part of antibodies, some are part of surface receptors, some are bacterial adhesion proteins. I think some of this is probably interpolation, interpolation here instead of extrapolation here. But we need to do more to figure out how much of one is going on versus the other. And I think maybe we.
Just continuing on that question, is the UMAP done on the embedding space, on the full embedding space?
It's done on the full embed. Well, let me catch that. It's done. So the embedding space is for these. So the question is whether UMAP is done on the full embedding, the full kind of latent representation that one of these models is capturing. So maybe, depending on which one you're thinking of. So they are done on the averaged embedding across all sequence positions. So the full embedding is a matrix, 1280 by whatever the length of the sequence is. And these are done on the 1280 dimensional vector that you get averaging over all sequence positions. So there actually is more kind of richness there that's not going into the embedding, which is just a choice having to do with data storage and compression and actually doing those computations. I suspect, I'm not going to put Zach on the spot like right this second, but he might know more about sort of how much going between the averaged and the full kind of representation changes the projection that you see. But we use the averaged one for this. Anything else? Can I just cut this myself then?
All right, let's give our speaker one last minute.